let's call on the third presenter, Bridget M. She'll be speaking on SPHERULE remodeling and endospore development in the fungal pathogen Coccidioides POSADASII. Bridget, are you here? Hello?
Can you hear me?
I can hear you. You can continue. You have the floor.
Excellent. So thank you for the invitation to speak today. I know we're all facing some challenges, and I would love to be in Prague with you all right now. I've never been to Prague, so hopefully we can make that happen in the future. So today I'm going to be talking to you about some recently published work. So if you are interested in more specific details on what I'm going to speak on today, the article is part of a special issue in Frontiers in Genetics on human fungal pathogen genomics. So next slide, please. So Coccidioides, the organism that I work on, is very interesting for this specific dimorphic life cycle. So the environmental life cycle, as you see on the brownish colored side of the picture, is most often associated with rodent burrows. The spherule structure development is initiated within a mammalian host at 37 degrees, and respiratory route is the most common route of infection. What I'm going to be talking to you today about is endosporulation. So this is a key step during infection. So this development of the spherule structure and the interior development of endospores, the rupture and release of those endospores continues the parasitic life cycle in the host. Next slide, please. So in my lab, we're very interested in spherule development. We know that environmentally it is triggered by an increase in temperature, so 37 degrees or above, increased CO2. These arthroconidia that are inhaled into the host undergo isotropic growth. Around 5 days, that spherule is mature and will release those endospores. Chitin synthesis and remodeling is a crucial aspect of the spherule structure. Approximately 96 hours, there's a formation of these endospores, as I said. And that rupture actually releases signals to the host and results in an increase in clinical symptoms like fever, etc. Next slide, please. So this stage is really critical to overcoming host defenses. That endosporulation formation occurs over a period of 96 hours. The endospore release occurs within that 96 to 120 hour time period. And this allows that fungal burden within the host to increase exponentially. Unfortunately, we know very little about the genes that are involved, both in the formation of the spherule as well as the release of those endospores into the host. Next slide, please. And this is just a histopathological slide of those two sort of phases. So on the right, you see a mature spherule that is full of endospores. And then on the left, you see that actual process of endospore release. So you see the cell wall breaking open and those endospores being released into the host tissue. Next slide, please. So a major gap in knowledge here is obviously what genes are utilized during this endospore formation and release. Could we potentially find druggable targets, right? So could we potentially block this particular phase in the host? So the work that I'm presenting today is the work of my PhD student, Heather Mead. And again, this is published research, so you should be able to find this manuscript pretty quickly. Next slide, please. So if we want to identify those transcripts expressed in spherule endosporulation process, what would be a strategy that we would take? So we have chosen to assess and compare transcriptomic profiles between a mutant strain that basically is blocked at a certain stage in this endospore cycle. So we know that chitin remodeling is a tightly regulated process in the parasitic life cycle. Individual deletion of those chitin-related genes doesn't really change the phenotype. So this is, again, published work from Gary Cole's lab, Zhu et al., 2009. They developed a lab strain that actually has the deletion of 2 of these chitin-related genes. And it's blocked in that intermediate stage of endospore development. So it creates an immature spherule. It causes infection, but does not cause disease in mice. And it was developed as a potential vaccine strain. Next slide, please. Okay, so again, as a schematic of what we're comparing here, right? So the triple knockout mutant, as we refer to it, is attenuated in the mutant, or is attenuated in the host, and it's basically blocked. So it forms the spherular structure, but it does not form endospores. Versus the wild type parent strain, which is the C735 background. So it completes the spherular life cycle stage, endosporulates, and causes disease in the host. Next slide, please. So we grew these strains under spherulating conditions, harvested RNA at 96 hours and mycelia for both of these strains and compared their transcriptomic profile. So on the left in panel A, you see a comparison of the triple knockout mutant 96-hour spherular transcriptome profiles with the 96-hour mycelial profiles, resulting in a comparison. So this traditional volcano plot, right? So we see the significant genes that are differentially expressed between spherules and mycelia. And we can complete the same analysis for the wild type, right? So then we have all of the genes that are upregulated in that spherular phase, both in the mutant and in the wild type strain. Next slide, please. Right, so then we can take those highly expressed and significantly different genes and compare them. And commonly we use a Venn diagram to compare these expression profiles to get a set of genes that are differentially expressed between mycelial and spherules for each of the strains, and then compare those. And this resulted in a list of 222 genes that are highly upregulated in wild type spherules, but downregulated in that triple knockout strain. So what are these genes? Next slide, please. So of course, many of these are very specific to Coccidioides, and so we don't have a known function. So about half of the genes we had no known function, but for those genes that we could determine function for, it appeared that nitrogen utilization was an extremely important aspect of this class of genes that were upregulated again in that wild type. So representing genes that are important for that endospore development. And I won't belabor this because I can see that this table is actually not very easy to see on this. So next slide, please. So another thing that we can look at is a group of genes that are improperly downregulated in the mutant. So we discovered about 19 downregulated genes. And so on the left-hand side, you can see that comparison with the mycelial phase and the, the left panel there in panel B is looking at the expression profile for the mutant. And on the right-hand side is the expression profile for the wild type strain. And it's looking, the first column would be the expression profile for mycelia. The next one would be the expression profile for the spherule. So that's the immature spherule that is not properly forming. And then again, mycelia and the rupturing spherule, so the proper regulation. And what you can see for those 19 genes is that in the mutant, these are basically improperly regulated. So they're highly upregulated in the mycelial phase when they shouldn't be, and downregulated in that spherular phase when they should be upregulated. So these are an interesting group of genes. Many of them, again, are hypothetical, but there's some heat shock proteins, there's catalases, some mitochondrial associated gene, trehalose genes, alcohol dehydrogenase, some genes that we know in other fungi are important for pathogenesis. So this is an interesting group of genes to further investigate. Next slide, please. And then again, conversely, we can do the same comparison for those genes that are potentially highly upregulated in the mutant spherule when they should be potentially off. And we found about 18 of these genes. And again, a large number of hypotheticals with domains of unknown function, but there are some proteases and major facilitator superfamily proteins that are present. But the story was a little bit less clear here. So again, some work to complete to really functionally characterize these genes. Next slide, please. Oh, whoops. It looks like that came through a little a little unclear. So what we did to further really understand that spherule specific is, rather than doing a comparison of upregulated or downregulated between spherule and mycelia, is to directly compare the transcripts of the mutant spherule with the wild type spherule. So this is just comparing mutant versus wild type genes. And so again, that same comparison that we did before, but not looking at a comparative, just looking at the direct transcript profile. What we observed is there was only a single gene that was highly expressed in the mutant that was not highly expressed in the wild type, and that turned out to be the resistance cassette that we use for the knockout. So the hygromycin gene, which is used as a marker for deletion of the chitin synthases, chitinases, was highly upregulated. But there was a series of genes that were there was many more genes in highly expressed in the wild type that were completely off in the triple knockout. So again, this is an interesting set of genes that we would like to pursue further. Sorry for the error in the slide. Next slide, please. So this one looks like it's not showing up for some reason. So I'll try to describe what you should be seeing here. So basically, there were two interesting genes that popped out of that analysis, right? So these are genes that are highly upregulated in the wild type spherule that are basically completely off in the mutant spherule. And they happen to be genes that are co-localized in the genome. So if we look at the genome, we see that these genes are right next to each other. And there was an interesting expression profile for these two genes. And what we observed is that it looks like a molecular switch. So these genes are so one of the genes in the wild type mycelia is expressed, but very low, and then more highly expressed in the spherule. And then if we look at the gene that's right next door, that expression profile is flipped, where we see that expression is higher in the mycelia and lower in the spherule. So hopefully we can see that pattern on the next slide. So we were able to also understand expression of these genes in a murine model of infection. So we use an approach where we infect mice with Coccidioides and extract RNA from lungs primarily, and then use nanostring to actually directly hybridize probes to RNA molecules of interest. So we get away from cDNA synthesis and any sort of issues that might arise from that. And what we observed in vivo is that, so again, some control genes in those first sets of panels, and then the CP37 6600 or 660 and 670. So we see the same expression profile where the one gene 660 is very highly expressed in vivo, and that's the one that is associated with, we believe, a switch to forming that particular spherule structure. And it's the neighboring gene in vivo is also downregulated, which would be the prediction that we had made. So I think I will close there. So hopefully the next slide is my thank you slide. Yes. So again, I have a team of folks that help with all of this work in my lab. Primarily the work for this particular paper was led by Heather Mead, my PhD student, but Claire and Marley also assisted with some aspects of this paper. And then final slide, next slide. So a lot of acknowledgements, so funding sources, NIH, some state of Arizona grants and some foundation grants, and a lot of past members and a ton of collaborators that make all of this work possible. And I think with that, I will close and take any questions.
Thank you so much, Bridget, for that amazing presentation. I believe we have questions online. Okay, are you ready for your questions? Is infection also linked to the microbiome of the skin?
So this is primarily a respiratory pathogen. So I don't believe so, but yeah, the microbiome of the lung is, and that's work that's ongoing.
Okay, thank you. Another question, because we still have time. Do we really know the entity, the entire life cycle of Coccidioides, or are there some hidden stages?
There are absolutely hidden stages. Another project in my laboratory. So that dimorphic life cycle represents only the asexual portion. So that's all asexual clonal development. There is clearly a sexual life cycle for this organism. And another PhD student in my lab, Claire, is working on discovering the life cycle. We've actually we're very close. We have gymnothecia. We believe we have ascospores. So now we're just trying to figure out how to deal with those ascospores in the confines of the biosafety level that we have to work at. So it's been a little bit challenging to harvest viable ascospores, but we are very close to that. So hopefully in the next year we have that publication out and we can share that life cycle with you.
Okay, thank you so much, Bridget. Wishing you good luck in your future research.